I can introduce myself. My name is Rudolf Merkel. I'm glad that I'm not introduced by some chairperson, because they tend to tell you all about your personal history, and then everybody realizes how old you are. So you can rest assured I'm old, and yeah, I will speak now about mechanobiology of cells. This talk has been prepared during the last nights. When I came here, I thought I would give a review talk about everything, and then I realized these guys really want to understand, and they ask a lot of questions, and so I didn't feel up to defending other people's work where I maybe don't know all the details. So I'll cover some basic principles, but I'll stick in broad parts to what we are doing in the lab. And yeah, therefore it's a personal view, and the selection is to some aspects arbitrary. Actually, I'm very happy if you ask questions, because yeah, you know, in Alice in Wonderland, the king tells her to start at the beginning, tell her story, and end at the end. But this is a story without end, so we can end somewhere, and if you have asked a lot of questions and we have discussed and everybody has learned something, I think we have done our job. So, okay. The first thing I would like to yeah, if this thing here works, yeah, I would like to draw your attention to is that all organisms react to forces. You see here an example, quite a lot of dynamics here, and I've selected that picture because it somehow characterizes the state of the field. It certainly is dynamic. There's certainly a lot of action going on. It's certainly also fun to work in. But yeah, as you can see here, it can be extremely messy. So yeah, the problem is, on a scientific level, there is a lot that is messy and not yet understood. Okay, so also mechanobiology is a word that needs some explanation. There are two flavors if you want to study biological systems in the context of mechanics. Either you come from physiology and mechanical engineering, you are looking at continuum length scales, you want to understand why some guy is able to run faster than you, and so on, then you call that field biomechanics. Now other people realized this field is fun. These guys, including me, come either from molecular biology, that's not me, or biophysics, that's me. And we think a little bit different, but we also work on biology and mechanical force, and to stress that everything is different, we call that field mechanobiology. It's essentially the same thing, just worked on from different angles, so don't worry about names. The basic effect that's going on is some force impinging on here a cell. That's actually how a physicist would see a cell. It's just some squishy bag with some material in there. Then you must have a first sensory process occurring in some molecule. So you must have a primary sensor for that force coming or that deformation, otherwise you wouldn't have a reaction. Afterwards, you have a secondary signal cascades. You've seen these frightening schemes of chemical reactions of thousands of molecules acting on each other back and forth and feeding back several times during this seminar already. And yeah, in the end, the outcome is a reaction of the cell or the whole organism. So a signal is basically worked on, transformed into a reaction. And that's what we are looking at. Now, there are many sensory mechanisms that might be happening. There is mechanosensitive ion channels, so force creating an ion influx, and you've seen already calcium is playing a role in almost every cell signaling event, so that could be an effect. You have mechanical straining of molecules. You've seen also examples of that type, where the straining, the basically opening up of a molecule would liberate some cryptical, usually hidden kinase target sites or some enzymatically active site. Or you have conformational change of a molecule. Basically, the mechanical force is just pulling it into the right geometry for another molecule to bind. All these might be events that result in sensing of a mechanical force. And the last effect that's also there, you just rip apart something, and these breakdown molecules might be bioactive. Okay, and to realize there are many hypotheses out there. A lot has been proposed, but very little, or in rare cases, you have a good proof for something. Yeah, please.
Can you give me an example of this mechanical brick of structure?
I will come to one at the end. You will see something. Okay. Okay. Now, as life is difficult, and you've seen there is a lot of complication, and I'm a physicist, so we are taught to, in our lectures, to look for the first and last things. So I'm really interested, yeah, we are the theologians of science, and so I'm really interested in that sensory mechanism. And let's look at an extremely simple system, that's bacteria. That system is so simple that it's fairly well understood and proven. In a normal bacteria, you have basically a sausage, a bag, that's filled with cytosol. It has a high osmolarity, and the outside world is something. Imagine the bacteria on your teeth. If you drink pure water, they are bathing in normal water. If you drink a beer, they are bathing in something else. And so the outside world has very variable osmotic strength. Now, this difference in osmotic pressure must be balanced by something, and what's balancing this pressure in bacteria is a mechanical stress or force, tension in a shell. This shell is depicted here. You have a porous outer membrane. Because it's porous, it's not a barrier for osmotic effects. You have an inner membrane that's impermeable to the solutes, so the osmotic pressure is acting over that membrane. And then you have a peptidoglycan. It's basically a cross-linked polymer layer. It's like, some bottle of, of some detergent or so. And in the end, this peptidoglycan layer is a cross-linked material. It's a resin, and that can take the force. It's like the layout of a bicycle tire. You have the outer tire that's there to withstand the force, and you have the inner tubing that's there to hold the air. It's the same layout here mechanically. And now, to study that is pretty straightforward. You have a bacterium at low osmolarity. The cell is in balance with its outside world. And then you bring it into high osmolarity buffer. They call it hyperosmotic shock. Now these cells will shrink. Water will diffuse out of the membrane because the osmotic pressure difference. Cell doesn't like it, so it regulates against it. It will increase its inside solute concentration until it's in balance again. Now, experimentalists can be pretty mean guys. There is what you can do. You take that bacterium and throw it in the distilled water, basically. Now, the inside will swell and load the outer shell with a high mechanical force. And if the osmotic pressure shock is strong enough, you have two possibilities. If the cell cannot relieve that osmotic pressure, it will just explode. Bomb, it's gone. Or you have some basically safety valve that make channels in the membrane that will open. The cell will sacrifice some of its solutes, but it's better than dying. And after these solutes have left the membrane or this sausage, the osmotic pressure balance is back to normal. The gates close again, basically, and the animal or the bacteria lives on. Now, this is a very simple assay, and it's often forgotten how important it is to have simple experiments. I learned that from Paul Hensma, who introduced AFM, atomic force microscopy, into biology. You have to build a setup in such a way that you can do many mistakes a day and not only one a month. Because you want to find out new things, and so you have to test a lot of things. So, and therefore you need a simple and straightforward assay. And that's one. That's one of the reasons why this bacterial system is so well understood. It's an easy experiment. Now, what people have done is all the molecular biology. They have localized which molecule is responsible. They found out how to clone and overexpose that, and how to reconstitute that in model membranes. This is the so-called large conductance mechanosensitive channel MSCL. This is reconstructed in a model membrane here, and that model membrane can be sucked into a glass pipette, a micropipette, and then you have full control over the system. You can apply an internal pressure that will change into a tension of the membrane, and you can also measure how much current you have through the system. You can measure electric current through that membrane, and so you have a signal that tells you how many of the channels are open. You can directly measure mechanical tension and the response of the system to it. That has been done by Sukharev, 1999, in a series of papers. Here are the experiments, pressure in millimeter mercury, so torr. And you see the membrane is pretty taut, and from the curvature and the pressure difference, he could calculate straightforwardly the tension. And what he gets here is a current versus tension curve. And you see with each step in suction pressure, he increases step by step by step the current through the membrane, so the gates open. That can be, this is plotted here, fraction of channels open. You see it's a biphasic behavior, and at a tension of about 11 millinewton per meters, all these gates open.
It's a huge tension.
Yes, everybody who ever worked with model membranes in the lab will be afraid to work with that system. I did that, and it's pretty painful. And so this is very, very close to the lysis tension of a membrane. Actually, I guess one of the major problems in that study was finding a lipid composition that would last long enough to withstand these 11 or 12 millinewton per meters, and that's also the reason why here are no further points, because down there you will just rip apart the membrane by itself. And that also tells us this is a last-ditch measure of the or action of the cell. It's either opening those gates or exploding. Now, how to understand that? The channel itself has two states. It's either closed or open. And as it's a chemical reaction, you can describe that in the standard image with a free energy, or for the Germans among us, free energy is not freie Energie. It's just the chemical, the thermodynamical potential that fits to the problem. Okay? Don't mix it up. So it's, in this case, it's Gibbs free energy. Then you have because the gate is closed, usually the closed state must have lower free energy compared to the open state. Now what you do when you pull on that membrane, you can deliver energy to the system, because in the open state, the footprint of this system in the membrane is larger. Okay? What adds to that is basically you get an additional mechanical energy term. And that's simply the product of membrane tension, this is energy per area, and the area change. And that change feeds into opening. And so this usually disfavored state is lowered in free energy until it's lower than the closed state, and then basically the gates open and the system relieves its osmotic pressure.
What is important here, the cut of the energy barrier, or really the decrease of the basic energy? What was
as far as I know, in this system, you're raising a good point. Basically, in this system, it looks like, if you look at the data, that it really comes to equilibrium. So you can calculate straightforwardly from this one what you would expect as fraction of open channels, and the system seems to reach that. So the barrier can be overcome. There can be also systems, if that barrier is extremely high, the whole process would be kinetically hindered, and then it's important that you lower the barrier.
Which also happens due to the same...
Certainly. It's the same mechanism basically. It's just you have a mechanical contribution to the free energy, and that will basically change the balance of your states. Yes?
But can I just check, you're saying that the change in state stems from a mechanical process rather than a chemical one?
Well, the transition, this is basically like an isomerization of a molecule. And it is coupled to a mechanical force. You have just an additional term, tension times area, the usual thing to add to the free energy terms, intensive variable times extensive variable that act on each other. And this term only comes into play if you have a change in area. Otherwise, there is no coupling. For this system, MSCL has by now been solved in its structure in 2 states, and there really is a change in area. It's not only a hypothesis, it's there. Okay. What I want to point out, it's a mechanically driven chemical reaction, and it's therefore by itself, it's a stochastic process because it's a single molecule undergoing a chemical reaction. That's statistics. Now, this is just the calculation, how it's straightforward. It's just principle, I think it's physical chemistry 2 or so, what you would expect. And this fits the data pretty nicely. You have a usual free enthalpy in the starting state, and most importantly, you get a measure for the area change that must be there in order to lower this barrier or this second state to the required low level. The kinetic analysis shows that there are substates, so my picture here is a bit too simplistic, so there should be some bumps in here, but it's not important for the principle. Yes?
Do you need to pull in more than 2 directions in plane to have this?
More than 2 directions. Membrane tension in model membrane is a 2-dimensional fluid. It has no shear resistance, so membrane tension is always isotropic. So you have always in 2 dimensions the same force per area. Okay. Now animal cells. Animal cells are pretty much more difficult. And therefore the situation there is far from clear when it comes to mechanosensitive ion channels, because they are most likely almost tethered to either to the internal cytoskeleton or to the external matrix or to both. And the reason is quite straightforward. Mike Sheets and his group have measured membrane tensions of animal cells a while ago, and their typical values are on the micronewton per meter range and not on the millinewton per meter range. If you inflate eukaryotic cells like dictyostelium to such a level, they will just explode. The membrane is too unstable to sustain such high tensions. So, and if you take such tension values, then you would need an area change in the membrane of 1,000 square nanometers in order to get a reasonable energy contribution to switch. And that's, yeah, it's fairly inconceivable that this would happen. No molecule could do that. And therefore, you need a mechanical coupling to something in the 3rd dimension that would basically pull on that channel and open or close it. Such channel exists, for example, in the hair cells. Usually I speak about that in my lecture in German, therefore I sometimes fall into not remembering a name. Thank you. Okay, so in some cases these channels are proven, but in general for animal cells, mechanosensitive ion channels are a pretty dicey business if you want to prove it. Okay, and the problem there is they are part of large multiprotein complexes, and therefore, if you would like to purify them, to do experiments on them, you would have to reconstitute the whole complex, which is close to impossible. Okay. Now, the same principle in essence can be applied to unfolding proteins. You have a folded protein, you apply a force to it, it can open. The distance between the ends, or where the molecule is attached to something, changes due to that unfolding. Basically, you just rip out that fold. And so you get a lowering of the free energy along the reaction path. Here it's more believed that it's more a lowering of the barrier and not so much a change of two states, but maybe this is a matter of belief, because the outcome in the end is the same. You get a reaction rate for this change that depends exponentially on the force you apply to the system. So you rip hard enough, that thing will fail. And then you have opened here, and all reactions can start. This model was proposed by Bell many years ago and worked out in great detail by Evan Evans. Not so long ago, but also a bit ago. Okay. Now, if you have such a process, cell signaling starts. I learned from how lively the discussions are here. I should not speak about things I don't understand. So I will not speak about cell signaling, because I keep mixing up protein names and so.
This will generate a discussion, otherwise it's boring.
Yeah. Oh, well, I hope what I speak here about will also create a discussion. Okay. Now, if you want to do mechanobiology, you have to be aware a bit of the structure of the system. This has been presented quite often already. You have a cell that has a cytoskeleton connected to the extracellular matrix at some adhesion complexes. We've heard a lot about focal adhesions. There are others also. I didn't attempt to be complete here. Also, my picture of the focal adhesion complex is a pretty old one. It's a book from 1999. So by now the pictures look very much more complicated, because many proteins have found to be in there. But don't worry about this. For the mechanics, it's just you have two networks or two meshworks that are connected. And these meshworks can look entirely differently. Here, extracellular matrix structures. This is from skin, so you have it's like plywood. Then you have also loose networks. This is a famous picture taken by Nishida et al. From fibroblasts in the rat cornea, where obviously the meshwork has to be very thin and fine and homogeneous. And if you have basal lamina, like here in the cornea again, then you basically have a two-dimensional, like a felt the cell lives upon. Now, the inside of the cell here, a scanning electron micrograph of the actin cytoskeleton within a fibroblast is also pretty complicated in structure and can also change. You've seen many examples of that already here. And in order if we work on such system, we have to take care of all these complications. So, and there many problems arise. Can we describe the outcome of our experiment by a continuum model, or do we have to look at discrete molecules at individual fibers and so on? We have to look at force transmission pathways. So if you pull on such a structure, it's not per se clear that if you take, for example, that fiber and pull, that you would pull on all fibers. If the network is loosely connected, it's like pulling a single fiber out of for example, of my shirt. And such things one has to worry about. Then we have stochasticity. We have discussed this already quite a bit, by the chemical reactions, by low concentrations, and also by the porous network structure. And what is puzzling physicists all the time is the molecular diversity. Okay, now let's come to the point. These are the problems. Let's come to the experiment. How are cells reacting to mechanical signals? The first signal I will speak about is stiffness. If you look at the human body, you will find a huge range of stiffness, from bone that's somewhere like glass, and down to brain, where you have the lowest stiffnesses at all. So the softest part of us is our brain. Sorry?
Yeah, it depends on the person.
Yeah, you're right. let's not go too deep into that. Okay. The other mechanical cue I would like to speak about is strain. If you consider your body and think about which part of your body is somehow moving, it become again back to the brain. It's basically the only organ that is not somehow in motion at all the time. Everything else is basically moved and strained and worked upon all the time, either by the action of the heart or by breathing or the peristaltic movement of the gut and so on. So the motion is everywhere, even though we rarely think about it and we rarely capture it in cell culture. Now, again, another complication. If you think about such a system, you can do experiments on whole animals. You can have a transgenic mouse and watch what happens if you knock out a molecule. But it's very difficult to interpret, and it's very difficult to apply a very clear-cut signal. So we need, or I need, to come up with some numbers and hopefully in the end an explanation. I need a very well-defined system, and therefore I also need a model. My model is not virtual in a computer. My model is a cell ripped out of the body and placed in some device in cell culture. And that's what we work on. And that allows us to define mechanical cues that cells experience. Oh gosh, this should be a light blue here. I'm sorry, the projector represents that not so well. So the idea is to apply stiffness as a mechanical signal. You take a cover glass and place something very soft on top. And on that you cultivate a cell. In my lab, we, for some reason, started to work with silicone rubber, which you can also produce very soft. Many labs work with polyacrylamide, which every biochemist knows how to handle, and there you can also produce extremely soft gels. So there you can produce stiffnesses in the physiologically relevant range, because cover glass or cell culture plastic has a stiffness of about gigapascals, and that's physiologically maybe for some bone cells, but not for other cells. It's way too stiff. Now, yeah, this is only what we use, Sealguard 184. You don't have to look at the chemistry. The idea it's a two-component system. You have basically a backbone material that you cross-link, and by just changing the mixing ratio, you can go to very low cross-link density, then you get extremely soft materials. At the moment in the lab, we are down to 300 pascals. The hassle to work with such materials is if you usually touch silicone rubber, it's relatively rigid and decent material. These extremely soft silicone rubbers are extremely tacky. In one of my students was frustrated at some point and threw such a ball of this thing to the ceiling, and that hangs there since 2 years. So it's impossible to get it down. That's really tacky. Okay, what happens if we expose cells to such things? So here is one example. The most famous one is from the Engler group or the Discher group. Back then Adam Engler was with Discher. They could show that the stiffness of the substrate would influence in what lineage mesenchymal stem cells would develop. What we found in our lab is that if you plate cardiac myocytes on such silicone rubbers, you would see on softer silicone rubbers, you would see mostly such roundish fried egg shapes. Cell is just there. And on stiffer substrates, you would only see such structures where you see by at least if you go out camping, you recognize immediately you have anchor points and then you have taut ropes in between. So these cells are spread out, spanned, and under tension. So there is really, and this is, you just plate them on another stiffness, you get different shapes. Okay, another extremely famous example and very impressive example is fibroblasts from embryonic rat heart. If you plate them on a very soft rubber, 1 kilopascal, you would see such immunofluorescence images. This is a smooth muscle actin that they are filled with. And here the red signal is a vinculin that stains focal adhesions. And a single cell here is, for example, that structure is a single cell. If you take the same cells and you plate them on a more rigid rubber, here we used 16 kilopascal, then one single cell is this whole beast. You see that the smooth muscle actin forms huge, thick bundles, and also the adhesion structures are extremely large. They are super major. So it really looks like an entirely different cell, and in some sense it also is, because that stiffness signal is enough to induce differentiation of this cell type from a normal fibroblast to a myofibroblast, and that's irreversible. If you take these cells, trypsinate them away, and plate them on 1 kilopascal, they will still be like that. That has been published by Tomaszek and Gorin. Okay, and the switch is at a stiffness of 12 kilopascal, which is incidentally about what is given as stiffness of intact myocard. So this system is certainly adapted to the human heart. Okay, another mechanical signal we enjoy to apply is strain. We basically build the rake for cells. What you see here is a silicone rubber cell culture chamber. It can be strained. We mount it on these holes in a stretching device, and when you move the device, basically you pull on that structure or on the thin lamella that forms the floor of this chamber, and that's where the cells live. So what these cells then experience is it's basically like pulling the carpet under us. And yeah, there are several groups who've done that already. We are also working along that. The oldest work I found was of Starch and Bates in the 1980s. And what they did, they cultivated endothelial cells, a confluent layer, strained the whole silicone rubber sheet along this direction, and what they find is unstretched cells have some preferred direction, are roundish or somewhat elongated, and after straining they are more like elongated, and they said it's mostly perpendicular to the strain direction. Basically these cells reorient to evade the stress. That's the whole story. Now
these cells were plated after the stress was applied, or
no, they were plated, waited until they are adhered to the substrate, they were cultivated to this confluent layer basically, and then the straining was begun. Unfortunately, they had a device that worked with 1 Hertz, and 1 Hertz, 15%, but 3 days of cyclic strain. Yes?
Are there junctions between these cells? Are they mechanically connected?
They didn't show it in their paper, but I'm sure they are.
what would happen if one plates cells on a stretched substrate?
Good question.
Instead of rigid substrate. I mean, there should be something of common. I mean, whether the cell senses stretch in the substrate as it senses rigidity.
Okay. So your point is, your question is basically, if I take a rubber sheet, strain it along one axis, and after that I plate cells.
Right. Whether they would sense the stress in the substrate.
Yes. Okay. We've done that experiment, and we had 2 outcomes. First, it's interesting. Sometimes you learn most of the artifacts you produce. First, we got a result. We got that cells would align mostly along the stretch direction. And then we found out that the reason was extremely simple. Because these cells don't adhere to the naked rubber, but we have to coat it with an extracellular matrix protein. Like we most often use fibronectin. You could also use collagen. And that molecule is then deposited. If you now strain the substrate to a large amplitude, you would align these fibers. And what the cells recognize is the alignment of the fibers. So if we what we did wrong with, we stretched a coated substrate and plated cells then. Now we stretched first, coated the substrate in the stretched state and plated cells again, and now the orientation was random. My idea to that is that the cell can adapt to its outside environment, and so if there is no change, the cell will just basically fall on that substrate and adhere and be in a happy state. What the cell feels is not absolute stress, but changes of stress and strain. There is if it's slow enough, the cell will just move around and not feel that the carpet under it is moved. That's my idea to it. I know it's anthropomorphizing what a cell would do and so on, but it's an idea.
In other words, the cell senses rigidity, but not the stress.
I wouldn't.
Rigidity determines the change of the stress.
Okay, if you think like that, but if you have the identical material, I think I should show you some more slides, and then we should come back to that. Okay. What has been shown is that if you have cells more separated, the cell body realigns. Then you can also study the dynamics of this reorientation. That was done by the Schwarz group or by Kemkammer in Stuttgart. They studied how fast the orientation would go if you switch on the signal, the cyclic straining, how fast the orientation of the system would decay after you switch it off. So there's quite interesting dynamics in there. And they found that the reorientation rate depends on amplitude and frequency. So the more amplitude you apply, the faster, and the faster you have the cycling, also the faster the reorientation is. Okay, then what also happens is that within a given cell, actin bundles will realign. You will see later on again an example. And now I come to my own experiments, or to our experiments. I didn't do it in the lab. I didn't have to suffer through that. What you see here is human umbilical cord fibroblasts after cyclic strain, always an overview image, large scan area and a single cell down here. Strain direction is horizontal along the double arrow. In control cells, we see all directions. After 8% strain, well, you see some weird guy along strain direction still, and at 32%, we rarely, if ever, find a cell that's still aligned in the strain direction. They all rotate away. And if you look at the individual cells, you'll see here, for example, stress fibers in this direction, in that direction. You'll find all directions. And here we find stress fibers only out of the strain direction. Now, if you think a nice picture is enough, the story would be over at this point. As a physicist, I'm trained to come up with numbers and statistics and so on. And therefore we devised a technique, or an image processing technique, to measure orientation of cells and orientation of stress fibers. The idea is very simple. There is a technique called the structure tensor that measures the local orientation in the gray value image. And together with some thresholding, we basically determine for each pixel what the orientation of the gray value gradient is in that environment. So we get here basically a lot of numbers that tell us directions. And that gives us a histogram of orientations of the fibrous structures within a cell. Actually, I forgot to mention, this is always labeled against actin. Now, it's a huge pile of numbers, and we have to characterize it somehow. We use the maximum of that distribution and say this is the angle of the cytoskeletal orientation of that cell. And the orientation of the whole cell body is very simple. We have, again here, we have a thresholding, and we basically calculate the second moments of that structure, and we use basically the ellipse with the same second moments, and that again has a direction of its major half-axis, and that's for us the direction of the cell body or the cell shape. Okay, now these are cytoskeleton orientations. You see here, this is the control and 5%. We come up with some thousands of cells that we study, so you get statistics with this technique, and you see at 5% strain you have a small effect here, at 8 it's very clear, and at 32 you have a huge effect of reorientation. Now, what bugged me is that this maximum was not at 90 degrees. Here you could worry, maybe it's statistics. Statistics is never perfect, even if you look at counting distributions and so on. There's always some other effect acting against you and randomizing your data, but still I didn't like it. Then, yeah, we looked deeper into it, and the reason is quite straightforward. Every one of you who has to teach continuum mechanics will know about that. If you stretch a material into one direction, it will shrink in the other. Imagine, maybe it's too smelly, so I will not do it. Take your sock, pull it. It will get narrower. This is exactly the effect that's happening here. And the ratio between elongation and shrinkage is the so-called Prussow ratio, and it's a material constant. And for a very soft rubber, it ought to be 1/2. It's a volume incompressible material. And we measured that, and as accurately as we could measure it, it was also 1/2. I know if you discuss that with somebody from mechanical engineering, he will worry if it's 1.50000 or 1.49999. It makes a difference for them. We couldn't measure that exactly, but it's almost 0.5 to the accuracy we can have. Okay, now if you now look on top of this stretched substrate and you imagine you draw lines, and then you measure how long is that line in the relaxed state of this chamber and in the extended state. You will certainly find an extension along strain direction, and because of Prussow ratio, you find a shrinkage in the perpendicular direction, and somewhere in between, you have an angle where you don't have any length change. Okay, very straightforward, simple geometry. Yeah, now I'm a stickler for accuracy. We also measured that in our chamber, and it turned out by imprinting a regular grid into that chamber bottom, and it turned out that here we wouldn't have 0.5, even though the material has a material constant of 0.5. We just didn't have enough shrinkage in the perpendicular direction. And the reason is very simple if you think a while about it. This chamber has side walls, and those side walls are stiff. The chamber bottom has to bend them. And those exert forces, and those forces basically hinder the shrinkage. You have a biaxial stress rate, and what we measured is a shrinkage ratio of about 17%. Forget about the last 2 digits, it's overdone, it's 17% about. Now, again, same calculation, you still have an angle of zero strain, but it's at higher values. And if you change that ratio here, you will shift that angle around. That's the important message. Now we did the experiment. We built this is the result from before, normal chamber. And then we had a chamber where the side walls were removed. So the shrinkage would be less, would be higher, and the hindering less. So we have here now a shrinkage ratio of 33%. And you see the maximum has shifted to lower values. So under these conditions, these specific cells basically reorient into the direction where they don't experience mechanical strain. There has been, unfortunately, Sam Safran is already gone. He has made a beautiful theory to that. The idea is, I try to sketch the idea. I wouldn't be able to reproduce that. It's really Sam's work, and I think he did a beautiful job there together with his students. The idea is, this cell is a forced dipole, and it has a preferred state of mechanical either stress or strain at its apices. Now, what the system, and this is a static image, but here we do a cyclic strain. If we just hold the stress, as I've explained, we don't observe anything. So what basically the system senses is the energy deposited to it with respect to its preferred state. So basically the cell goes to its preferred state as good as it can, and there is still wiggling around that, and energy is deposited to it. And basically that energy, Sam modeled as a free energy potential that would basically, in which these cells would reorient. It's like you have a magnetic dipole in an external magnetic field, and Brownian forces acting against perfect orientation. And in this case, he said, well, all the beautiful biochemistry adds some stochastic forces, and that's my temperature in this case. And yeah, this is published by Dea and Safran. They published many papers about it, but this Physical Review E paper is complete, and it has all the equations. I found it the best to read. Then later on, last year, Livni published an extension of that theory where they took into account that both the cell and the focal adhesions have a certain width. They are not needle-like structures. This gives the modification. Anyway, with Sam, yeah?
The energy dissipation is minimized or maximized?
Minimized.
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 Is there a physical rationale?
Is there a physical reason for it?
I mean, this is a postulate.
It's a postulate, actually.
In some sense, once you admitted this postulate, you put the answer.
Right.
In the
right.
It's no wonder that you get it back, the same answer. Yeah?
Yes.
Yeah.
Yes.
So the most interesting thing is what could be a mechanism for this, for sensing the energy dissipation by a specific mechanism.
Actually, now we are entering the world of hypothesis. Actually, you are perfectly right. Sam simply made up a rule and made his system follow that rule. And interestingly, with that rule, I could do a global fit to all these distributions. It has only one free parameter, and it looks fairly well. So I was really shocked that the fit would turn out so well. I never expected such a simple model to work out so beautifully. So it's not entirely wrong, but it's extremely simplified.
Don't claim that it's wrong.
Yep.
I'm trying to find.
Yep. And in the end, what's happening, or what must happen, is that we have a cell attached somehow to the substrate. We have this external strain, and now what's going on is you have here the mechanical connections inside the cell, and when you pull on it, some of the processes I've sketched as mechanical sensing happen. It's either that you break parts of the stress fibers, you open mechanosensitive channels or something. And it basically is a signal that feeds into the cell signaling. But how the cell signaling basically knows where its fixed point is, where it should be, I have no clue. I know it's a very vague answer and most likely not satisfying. But this certainly, if you have an answer to that,
No,
write a paper, write a beautiful paper.
I wouldn't ask if I would have an answer.
You'd be in your office and writing the paper, I guess. Okay, now an illustration of what's happening. This is a live cell experiment, a cell on this strain chamber, and every some strains or some cycles a single image is taken. This cell is transfected with live act, and so you can see the stress fibers, and because it's a transfection, you also have this artifact around the nucleus where the live act is also produced by the cell, and you will always have a central bright spot. But what you should look for are these fibers out there, and the substrate is strained along the horizontal direction. And I hope my computer will play this. Hello. Yeah. Okay. So what you see, maybe not so clearly because the focusing is pretty difficult in such a moving sample, but what one can see is that individual fibers break, others seem to slide and rotate, others seem to split, so that there seem to be all kinds of processes going on. Okay. Now we can apply the same algorithm as before to this movie, and what you see here is the same movie as before. Here you see where the stress fibers are found by our algorithm, and you'll also see the preferred, this is the histogram of directions of the fiber structures, and you'll also see that tiny yellow arrow indicates the direction of the cell body. What you find here is, come on. What you might have noted, it's difficult to look at 3 panels at the same time. The stress fibers rotate faster than the cell body. So you have, it's really the stress fibers come first and then the cell body follows later, which is kind of reasonable because the stress fibers are tensile structures and they have to take the force and the strain. So something is happening to them if you pull on the carpet. There's a question? Yeah?
Play that again.
Yeah, certainly. I like this movie, so I'll certainly play it again. Once more?
What is ICS 7?
Oh, that's my institute. Sorry. It's just that this is not yet published. It's Alexander Zielinski's work, a graduate student of mine. Sorry. Yeah?
The frequency in which you apply the cyclic strength, which is the value, is very low? Or?
Compared to most experiments out there in the literature, we used to pull at a low rate. I forgot which. Yeah, it's here. It's 130 millihertz. Most people tend to pull around 1 hertz.
Okay. If you change this frequency?
We changed that frequency, and yeah, thank you for reminding me. This ties back to Misha's question before. If you increase the frequency, if you look at these structures here, if you increase the frequency, you get more ordering. It looks like the potential is getting stiffer. And the same happens if you increase amplitude. So to some extent you can exchange the height of the amplitude for the frequency of the strain application. But there is certainly more to it, because Ronald Kaunas has done experiments where they changed all the shape of this oscillation. They use square pulses and slow rise, fast fall and stuff like that, and cells seem to react to everything. They even sensed if if stretching was fast or release was fast by mirror symmetric shapes and so on. So it's amazing. Yeah?
How long does it take, compared to the stress fibers reorienting, for the cell to then elongate in that direction?
It follows later. In this cell type, I don't have these here. In this cell type, I think we tested it at this condition, 14% strain. We had reorientation of the stress fibers was fairly complete after 3 to 4 hours, and cell body reorientation was complete overnight. So twice the time or 3 times longer. So it really follows later. But remember, what we plot here is the direction of the fibers, not the length. Okay. So what's happening down there are many processes, and I'll focus on the one we are currently working on, because I would not love to speak about things I don't understand. And then you'll ask me something and I have to admit I don't know. So what you see here is what we studied was a molecule called succin. Zixin is a molecule that is located both in focal adhesions and in stress fibers. What you see here, we needed 3 molecules, so 3 colors, and that's always nasty. The blue one never reproduces well, but what you see here is just an actin stain. You see the stress fibers. Then we had to label the focal adhesions. That's a stain against vinculin. And now we stain for Zixin. And what you see is, you see here that the focal adhesions are labeled, but also in the stress fibers you get foci. You get some clusters within the stress fiber. It's not homogeneous anymore. Before stretching, this cell was stretched just 1 to 35% amplitude. Before stretching, you get a homogeneous staining pattern. It's just that the stress fiber is homogeneously decorated. Now, then we went for a statistical analysis. So we have to distinguish what is focal adhesion, what is stress fiber. So we basically did a thresholding for Zixin, identified all Zixin clusters that we found, identified all vinculin clusters, and those are focal adhesions, and we have many artifacts there because it's always with staining. But in the end, we looked for those Zixin clusters that are not close to a vinculin cluster, and those must be outside a focal adhesion, so they must be in a stress fiber. And that's what remains after this logical connection of these 2 masks. Okay, now we can look for the angular distribution of those structures. What you see here is angle and a cumulative histogram. You see that the actin seems to be oriented. The idea behind it is very simple. Because we cultivated the cell on an unstrained sample, stretched it by 35%. If you have a random orientation of lines and you pull them by 35% in one direction, you will see in the end a preferred direction in strain direction. It's just a simple geometric effect. If you can calculate that back, and then this line is almost flat. And if we now look at those streaks within Zixin after straining, this curve is steeper than the actin curve. This tells us that most of those patches in the Zixin pattern are along stretch direction. So they are somehow connected to those stress fibers that take most of the load. Okay. Also, we looked at other molecules, well, I'm the physicist. I admired the beauty of those results, but I didn't do them. So you see here, again, actin in blue, Zixin in green, and VASP, another molecule that connects to these structures, in red. And if you have red, blue, and green at the same spot, you will see white. So you see here in the overlay beautifully that all these 3 molecules co-localize. So we have also VASP in those structures. Now, this is all beautiful. In immunostaining, you get beautiful images. The contrast is just great, but you don't see what's happening live. And that's something we really would wanted to see. And Alexander came up with a very smart idea. He again used the silicon rubber sheet on a glass slide. And now we have to basically pull one end of the cell and not the other in order to elongate a stress fiber. And that is done with basically a microscopic garden implement, a hoe. What he did is he cut a piece of a silicon wafer, glued that to some holder, put it in a micromanipulator, and then you can basically poke that hoe into the soft rubber and move it to one side. And because the rubber sticks to the glass substrate, you get a decaying displacement field here. We verified that with microbeads. It really looks like the expectation. So we are basically pulling that end of the cell more than that end, and so these stress fibers have to elongate. What you see here is now, or what you will see if my computer is mercifully to me, you will see here a Hubeck cell that is transfected with GFP6. Again, here the artifact around the nucleus, but what you should look at are these stress fibers here. Before, and the garden hoe is somewhere here, and will be pulled in that direction. And before straining, in this state, stress fibers are homogeneously decorated. And now watch what happens once Alexander pulls. Immediately you get these foci in zygzin. It's boff, it's just there.
You do it again.
You want to see it again? No? Okay, let's go back. Now come. Yeah, now it's again at the beginning.
Initially, how are they oriented?
The fibers?
Huh?
The fibers are that way. So, and the hoe is here. Certainly, Alexander selected cells that had stress fibers in the direction where he could pull.
Right.
Otherwise, he would have to rotate the sample and so on, which is a pain. Yeah.
What's the time scale for this? 20 minutes or?
The whole movie is 20 minutes, and you've seen, upon request, I've relooped it after 1/3. So this is not that much sped up. As fast as we can refocus, we have the foci. Okay? Now we looked also for zygzin, and he did the same experiments with zygzin and VASP. You see here, this box is around the focal adhesion. We use that signal to normalize, to basically calculate a way, or to normalize our images for photo bleaching. And here in this rectangle, there is one of those patches in a stress fiber. And what you see here are individual images taken after a certain time. It's those 20 minutes. And you see in the beginning, we have a huge signal in zygzin that decays away after a while. And we have the same in VASP. Here is next to it is a false color representation. In this red VASP channel, it's better to see in the false color representation. So we have, in both systems, we have a relaxation of those fibers. This is the time course if you calculate it directly from the movie you've just seen. These are the relative intensities of zygzin and VASP. He did do that experiment many times. And if it's now properly normalized, we can average together about 30 events. And you see the zygzin signal looks like this. Actually, I was shocked that we could fit this complicated process with a single exponential. This shouldn't be. But it's like that. So a single exponential fitted it, and the time constant is about 200 seconds. VASP, same effect. Time constant is about 150 seconds. And then we looked at a third molecule. It's CRIP1. And CRIP1, interestingly, goes away from those sites. The very moment you pull, it's gone from that focus, from that low streak, and it comes back within half a minute. Yeah?
Are these curves an average over some area, or?
No, actually these curves are. In this movie, we basically take this piece of image, this one, and we follow it, because you always have drift in these experiments. You pull on your sample, so you're bound to have drift. So the trick is to take the first image, then cross-correlate the next one with it to find where has our sample drifted. We do that in the zygzin channel. And then in all other channels, we average the intensity in this box. And that has been normalized so that the first image is 1, the last one is 0, and yeah, as you would do it normally. And then you can average them together. It's 33 of those, and here it's 29. So it's an average over some few cells. And the interesting fact here is that zygzin binds VASP, alpha-actinine, and CRIP1. Sorry for that mistake here. But only CRIP1 and alpha-actinine bind directly to actin. Now, the interesting thing is there is another group working on that, and to to my pain, they published a bit faster because they did the experiment in another way. It's the Beckerle group in Utah. And our experiments are on different cell types, so you cannot compare everything, but everything we did came out exactly as I did it, so I entirely trust their results. And they found that if they do away with alpha-actinin, the system still works the same. So alpha-actinin is not what brings succinate to the stress fiber. Our idea behind that is, and this comes now back to one of the first questions asked, if I can show you an example of damage. My idea what's happening is you have this actin cable, you pull on it, and what we create by a strong pull is there are certain nicks in it. We just damage that structure. And in order not to break apart, not to fluidize entirely, nature has evolved in order to bring some proteins there to basically repair that damaged region. I'm pretty sure this is a repair process. It lasts about some minutes. And what we are missing is basically the very first the molecule that is really the sensor for these open ends. That's still missing, but otherwise, you see here we have a pretty intricate dynamics. And that's, for me, that's an example where we destroy something, and obviously the open ends that form in this destroyed structure somehow release and attract other molecules. And that can be a signal for the cell to act on. Yeah?
And so somehow you're getting rebuilding of the fibers?
Yes. After
polymerizing.
Yeah, there seems to be.
But why then do you get rid of the fibers of the direction of the strain? This is a mechanism to preserve fibers of the direction of the strain.
Right. But it has a certain time constant. And my idea is, now you see that process lasts for some minutes. If the next strain comes too fast, and the next, and the next, and the next, in cyclic straining, these fibers will collect basically, they sample up lots of nicks, and at some point they just disintegrate. We tried the experiment, if you pull only once and fix the cells afterwards, you will not see this beautiful rotation. So it's just by collecting enough, and that might also explain why the system has a time constant. If you pull slow enough so the repair is complete until the next pull comes, the system just doesn't care about it.
Because if you had a model with no repair,
yeah,
and you simply said that the depolymerization rate increases with strain, which it does,
yeah,
then with time all of the actin will automatically align in the right direction because you'll have a constant rate of polymerization and an increased rate of depolymerization along the strain axis, which will get rid of it.
Yep.
And it'll give the same exponentials and things that you were seeing. The time constants may be wrong, but the structure of the model will be the same.
Yeah.
And so here...
Yeah. Actually, I'm pretty sure there's an infinite number of models that would produce such behavior. But therefore, it's important to look at all these different molecules, what they are doing to basically get the underpinnings. And so these are first steps, but the system is not yet complete. Actually, Anotida, how much time do I have left?
5 minutes.
5 minutes, okay. So that's maybe too much to enter a new topic.
Yeah.
So I'll acknowledge all my co-workers, all my present and former students. I have shown you quite some results that have been produced by students that have left the lab by now. Here in the background you see most of them are still in my lab. It's a fairly recent group image. And I also thank all authors of quality papers on mechanobiology. It's really, always I find a paper that really gives the details and tries to go to also the complicated aspects and not only the straightforward shiny explanation. I really love that, and I appreciate the effort these guys have put into their papers. And yeah, I also thank my fellow organizers for putting up with me and my a bit chaotic style. And I thank you for your interest. And those of you who want to read more, this is a small library, a small literature collection. Yeah, if you want to, I think we have a homepage where I can upload that.
Yes.
So you don't have to write it down. I would like to point you to one review. It's by Eichmanns, it's from the Chris Chen group. It's called The Hitchhiker's Guide to Mechanobiology. It's maybe not as funny as Douglas Adams' book, but it gives a fairly intuitive introduction into mechanobiology. Okay, and I'll be happy to take your questions, your ideas, and I hope I have provoked some of you to come up with good models that might be helpful for our field. Okay. Yes?
Rudy, why do you stretch the substrate always in one direction and not isotropically in order to see zixin binding filaments oriented in all directions? If you want to prove the idea that zixin, for example, marks stretched filament, it should be straightforward to pull isotropically.
It's not that straightforward to pull isotropically and to image. There are some devices out there. One is where you basically have a lamella and you press basically a ring into it, and that will stretch the whole lamella in all directions. And the other is you basically have a balloon and you inflate it. But with that one, it's barely possible to image because the whole system moves that much. As far as I recall, in all those experiments, they claim that the whole cytoskeleton basically dissolves within, if you strain with some few percent.
This is interesting.
This is very interesting, and the experiment we did with that garden hoe, it's difficult to do that in two directions at the same time. So this is obviously a preferred direction experiment. Yeah?
With respect to physiological processes, do you think that the kind of manipulation you do have any relevance or to migrating cell or to living tissue?
To living tissue, I'm pretty sure it has. If you... I should go back to one of the first slides. So people have done MRI on the heart and came up with strain values that the material in the myocard would experience. And that goes up to 30%. So 30% strain really happens in the body. And well, we usually work with some 100 millihertz, so that's a bit slower than your heartbeat, but certainly the peristaltic movement of the gutter, so that would be a fairly decent description of that or approximation to that. And also this strain chamber has a stiffness of about 50 kilopascal, which is also close to connective tissue or to basal lamina in many tissues. So it's not that far off. Yeah?
When you apply cyclic strength, you saw how the cytoskeleton reorientated. I would like to know if you have observed that the focal adhesion can be altered.
Yes, but what happens certainly is that in the end you have a totally different pattern of focal adhesions. So some must dissolve and others grow. We couldn't follow that online. It would be a cool movie to track the life cycle of a focal adhesion that is permanently kicked around basically.
So when you start, you have focal adhesion in one position, and at the end, they have changed.
We have another pattern of focal adhesion. If the focal adhesions that you have in the beginning dissolve and form entirely anew, or if they seem to slide over the substrate, I don't know. Looking at that movie of the stress fibers, what they do, because usually a stress fiber terminates in a focal adhesion or in a cluster of focal adhesions. I would suspect that we would find focal adhesions of all kinds. We would find some few that would stay, some that appear to slide because some of these fibers seem just to rotate, and we would certainly find plenty that would dissolve and be formed entirely anew.
What do you think?
But that's science fiction.
What appears first? The orientation of the cytoskeleton or the focal adhesion?
Would there be a time delay? I expect the processes to happen fairly simultaneously, but I've been wrong with my expectations before. So one really would have to measure it. Yeah, actually, expectation and cell work is a dangerous combination, I can tell you. Yeah.
So, following up on that nice question before, giving isotropic tension might be more difficult, but you could certainly alternate the directions of your extension. You have a square chamber, you could pull this way and then pull this way alternately.
Yeah.
And your mean lines of zero stress are going to be the angles, but of course no fiber is ever going to be unstressed. And,
You're right.
And that was a question whether you would try that, and whether the cells just give up. And then the second thing, which is interesting, is, I mean, given that you have a Poisson ratio of 0.5, in the sense from the non-biomechanics point of view, compression and extension are interchangeable if you have a conserved volume.
Yeah.
But these cells really don't seem to care about being compressed. They only seem to care about being under tension.
Possibly.
And that's, I mean, perhaps not surprising given how biopolymers and motors work, but it is interesting that you really do seem to be getting a response to being pulled, but then you're compressing the fibers in the other direction. That doesn't seem to make any difference.
Well, we would have to do the experiment. To cultivate a cell at a stretched substrate and release it and fix it afterwards.
Well, but just because of the Poisson ratio of your substrate, if you're pulling in one direction, you're getting tension and you're getting compression in the other. So you are, maybe not by your same percentage, but by some fraction of
you would get it, yeah.
your strain,
Yep.
you are in fact shortening the
certainly.
fibers in the other direction.
Certainly, that happens. Yeah.
And you don't see a response to that.
Nope.
So the crumpling is fine. So that means these fibers are basically in a regime where they're in the tension regime.
Yes.
Where they can adapt to being compressed easily, but not to being strained won't.
Actually, the image people have, or the picture is that you can also, some people have, I think it's on my list of citation, Colombelli et al. They use laser surgery to cut an individual stress fiber and observed how it would shrink away. And they come up with a picture where the stress fiber is under tension, and it also has a viscous element, basically, and it's connected to something that we don't see. So in the end, the idea is when you basically stretch the cell a bit and let it reside again, or come to quietness again, the tension in these stress fibers would build up again to some equilibrium homeostatic value. And when you relax the system, it's just simple, the stress fiber can shrink by those motor actions, or if it's too fast, it just can buckle in the third dimension. If you pull, you will create those nicks that I've shown you, and then a whole repair process has to start. That's the common picture at the moment.
But it's an interesting question. Your example of the bacteria, you have a tension sensor.
Yep.
There isn't a compression sensor.
Right.
So is that going to be a universal principle here?
I'm not so sure.
There's a tension. I'm not answering. I have no opinion. I'm just asking. Why is there an asymmetry of tension? Why would there or not be an asymmetry of tension detection versus compression?
Actually, I would expect that buckling is the most important answer. If you look at those sensing mechanisms, you open up something. If you have a molecule connected to some at its ends and you shrink it, it will not react as dramatically because it just can fold away in the third dimension. But for large volume changes or for large compressions, there will certainly be a sensor that in the end will react to the osmotic strength or the chemical potential of water or something in the whole cell. So there will be certainly a limit. Otherwise, cells might shrink to nothing. Yeah, please.
I guess
We already spent 15 minutes. Shall we postpone this into the coffee, tea?
No, whatever you like.
Yes.
It's your decision.
Yeah. Because it will, yeah. Almost run out of time.
Okay.
Yes, very nice discussion.
Thank you. Yeah, thank you for the questions.